commuted all the way from. Thank you. It's good to know. It's good to know. She did her PhD at MIT in the group of Rebecca Saxe, which is where Haley, our previous speaker, who did an incredible job of navigating the sound difficulties, came from in that seminar. Everyone remembers that. And Heather's going to talk to us today about her work with infant fMRI perception and knowledge. Thank you so much, Heather.
Thank you. Yeah, so I'm Heather, and I'm happy to be here. And I gave a talk here a couple of years ago, so a lot of this is going to be a repeat if any of you were here a couple of years ago. I'm just going to stand so I can see everybody, if that I'm not violating too many norms in that way. But I really like lots of interaction. So if you have any questions at any point, just interrupt me and ask. And I will probably try, if you all are very quiet, try to ask you questions at various points. But I'm eager to hear what you all think of this work, because you guys do a lot of infant work too, but just like studying different questions. So you're a good audience for me. All right. Oh, and it's mostly going to be work I did during my PhD, not my postdoc, because my postdoc has been focused on adults primarily. All right, so humans are capable of lots of really incredible things. Our brains support our ability to perceive the world, to think about ourselves and others, and to generate new forms of knowledge and art. But when we're born, we don't have that same capacity, right? These babies will grow up to be humans capable of incredible things, but right now they're not capable of a whole lot. And so the question that I have spent most of my career so far thinking about is, how do we get from infancy and what we know and represent in the world in infancy to these complex states of the brain? Or in other words, what is the initial state? And how do we change from there? And of course, I'm not the first one to ask these kinds of questions. People have been debating these questions for thousands of years. But I do hope that I can bring some new data to these questions by measuring responses in infants' brains. Right. So for my talk today, the first thing I'm going to ask is, can we improve methods to collect fMRI data from awake infants? Can we measure meaningful responses to visual stimuli in infants' brains? Do responses in temporal and prefrontal cortices emerge sequentially? Can we measure meaningful auditory responses in infants' brains? And then a little bit of future directions. Even though it sounds like a lot, it actually goes pretty quickly. So is there any questions? Are there any questions so far? Great. So first, can we improve methods to collect fMRI data from awake infants? All right, so you guys collect lots of fMRI data from infants as it is, but they're usually sleeping, right? But you can't measure responses to the world around a baby when they're asleep, right? You need them to be awake. And so when I started my research, there were only 3 published papers that used fMRI with awake infants. And I was really lucky to get into the Saxe lab, where Rebecca Saxe and Ben Dean had innovated a bunch of methods to be able to collect fMRI data from awake infants. And so I'm just going to review that for you. Now, some of you might be familiar with this coil, right? I know Ellen has a few. I don't know if they're still in use though. But basically what we do is we swaddle the baby and put them in a special infant head coil. They watch movies in a mirror over their eyes, and a parent goes into the scanner with them. You can see this baby's moving a lot. And so you all know that movement is really bad for fMRI, and it's also very uncomfortable for the adults going into the scanner with them. So this is a typical bold image from an adult. And I'm sure many of you have seen these kind of images before. But you can kind of make out parts of the image. So you can see that occipital lobe, the cerebellum, the brainstem, and the prefrontal cortex, and the sagittal plane. In the coronal plane, you can get some hints of the basal ganglia. You can see the temporal lobes. And in the axial plane, you can see the frontal lobes, optic radiation, and some white matter tracks and things like that. And so for you guys, you might be pretty used to getting similar types of images from infants, but with the methods that we used that Ben and Rebecca just started, the bold images are actually much more distorted, right? And so we used a sinusoidal sequence, um, and you can see the posterior part of the occipital lobe is really elongated. Sometimes parts of the brain are cut off, and it doesn't look like much of a brain at all. And this is a pretty representative image from what we got with that old scanner. And so then Ben and Rebecca, they collected data from a bunch of infants. I'm going to focus on the 8 participants that they collected data from while they were watching movies of faces, objects, and scenes. And the light purple shows how much was collected, but the dark purple shows how much was usable after removing all of the high motion data.
Would I be able to ask how you managed to get 120?
So that, yeah,
Which baby was that?
yeah. So the first 3 babies all had a parent in the lab, and the first one and the third one, the parents was Rebecca.
Oh.
So that's how we got so many from or they got so many from those, and then the second baby was a postdoc in the lab. Yeah.
Was she in there for 2 hours in the scan?
Oh, she was.
Rebecca was
They were
there
in there
for
for much longer. Yeah. Yeah. It's like a lot of time. Yeah. So yeah, they did a crazy lot of work to do this, and it's like not trivial. So that's why I like to but for us today, what I'm going to focus on is the response to faces and the response to scenes compared to the response to objects, right? So for us, it's very easy to tell that the faces and objects are very different and the scenes and objects are very different. But the question becomes, and we know our brains represent these object categories differently, but the question becomes, do infant brains also represent these categories differently, right? And so in adults, here's an example of what a face region might respond to, where it has a higher response to faces in purple than to objects in yellow. And in adults, this is what an example scene region might look like, where there's a greater response to scenes in green than objects here in yellow. So we're going to measure from those same regions in infants and see if infants' brains also distinguish between these categories. And so here are the responses to the same regions in infants. And you can see it doesn't look like there's any difference in response to faces and objects and no difference in response to scenes and objects. So that is confusing because if, most of you, I suppose, have interacted with infants, it seems like they would know that a face is not an object and that they're different. So you might ask, is this the true state of infants' brains, or is it a limitation of the method?
What's the age?
For their paper, I think it was 6 to 9 months. It was pretty young. Yeah, but old enough that you would expect them to know that there's a difference between faces and objects. Does that answer your question?
I'm just wondering whether we can say that from a page.
Yeah, like maybe it's too early, like we're measuring it too early, and if this is the true state, like their brains really don't distinguish faces and objects. It's total possibility. Yeah, that's the question. Any other questions?
Oh, I have one question.
Yeah.
If you were going to do recording in weaning infants at term, so like just after they're born,
Yeah.
would you expect any form?
I mean, I would, but I don't know. Would you?
I don't know. Like in theory, I guess like in other animals where they like open their eyes at birth versus don't, I think there's quite a lot of work looking at that.
Yeah.
All of the white matter is there
Yeah.
to be able to do this. So whether there's like a reason for that or not.
I mean, all the neurons are there, right? So they're responding to something. So the question we'll get to is like, what are they responding to? They have neurons in their entire brain. That's what a brain is made up of. And neurons represent the world around us.
Yeah.
So what is being represented in an infant's brain? So the question isn't like, are they representing anything? It's more, can we measure whatever it is and get a handle on like what drives activations in different parts of the brain.
I was thinking about, you know, when you pass a baby to someone that's not its parent, like the first time that the baby figures out that wails, and like the difference, like if you pass it a neonate,
Yeah.
they don't know.
Yeah.
But then quite early on with some babies, it's like a recognition of parent and non-parent.
Right. Yeah, that's very
where
interesting.
was going from.
Yeah. Go ahead.
Also a question. So for the typical face region, as the trust, it's more like the consensus about the specialized region in adults. But for infants, they may present.
Hold those questions for a little while. Okay. Is that okay? All right.
So smell might be one thing triggering. I don't know how connected the smell and the visual connection. If there's not the mother, but someone else, like father, knows it.
Yeah, so it wasn't always the mother. I showed you an example of it being the mother, but sometimes it was the research assistant, sometimes it was the father. It was just always an adult. Yeah. Anything else? Okay. So the first thing I tried to do is to improve fMRI data quality. And part of what we needed to do in order to do that is we designed some new headphones in collaboration with Sensometrics that are MRI safe and play sounds to infants. And then with my collaborators, Boris Cullen, his grad student, and Preet Gotra, we designed a new infant head coil. And this new infant head coil is size adaptable, so you can open it and close it around the infant's head and the headphones, so it accommodates headphones and different head sizes. And it also increases SNR by 25% relative to an adult head coil. These new head coils, this new head coil along with headphones also enabled us to use a higher resolution acquisition sequence. So with that old acquisition sequence that we were using, it was a sinusoidal gradient, which made it quiet and safe for infants' hearing, but also increased the distortion we saw earlier. That I showed you earlier, and the new acquisition sequence is like a more typical trapezoidal gradient, and it's much louder, but now we have the hearing protection, and we're presenting sounds, and we also used a lower TE to increase and a higher resolution sequence to help increase the quality of the data collected. And another thing that we did is before we started the scan, I would run behind the bore, play peekaboo with the infants to make sure they could actually see the stimuli in the mirror while I was measuring their responses. So just as a reminder, this is what the typical image looked like with the old coil. And with the new coil, this is what a typical image looked like. And so you can see it's much higher resolution, and you can actually kind of make out some of the structures that you would typically see in a brain. So that's exciting. So we're getting better data, but the question also becomes, can we get more data? Because we're not going to pop out a baby every year just to be able to collect data from it. And so here I'm going to plot on the x-axis, each participant, and on the y-axis is going to be the amount of data collected from each participant, and they're ordered by age. And so you can see there's like a variable amount of data that was collected from each infant, but it's not correlated with age. And then here, and I'm zooming in on the 37 infants that we actually collected data from, that we collected usable data from, and now I'm plotting in dark purple the amount of usable data. So you can also see that there's a variable amount of usable data. We used over 300 hours of scan time, and the amount of usable data we had was about 18 hours of data. So there's a huge amount of effort that goes into collecting these data, which you guys have already noted. But it's really exciting. So I hope that I convinced you that yes, we can improve our methods, and we can also improve the quantity of data we collect. And the new coil decreases the distortion and increases the resolution, but it's still really, really hard to collect data from awake infants. Are there any questions on this part? Any more questions I see to say?
Did you notice a difference between mum and dad going in the scanner with baby? Or were they both,
No.
like maybe didn't respond better to one parent over the other?
I didn't notice any in that way. Like some parents themselves were more comfortable in the scanner environment than other parents. And that was a better, I think, predictor is how comfortable the parent was in the scanner environment. But I don't have data to back that up. It's just like intuition. Yeah. Anything else? Okay. So can we measure meaningful responses to visual stimuli in infants' brains? Okay, so hopefully we're going to get into these perception questions that you had. So I'm going to talk about two different kinds of perception, person-centered perception and object-centered perception. And so these are two images of horses. The one image on the left is more complicated than the image on the right, which is a line drawing. And so the 2D representation is going to encode the differences between these images, whereas the 3D representation is going to treat these images as the same. So what that means is 2D is going to say, oh, that horse is brown and has hair and black legs, but that horse is just a line drawing, and the 3D representation is just going to be, oh, that's a horse. And so if you measure responses in adults' brains, these images are going to produce really different responses in primary visual cortex at the back of your brain, but they're going to produce similar responses in ventral temporal cortex. And it's nothing special about these two horse images that I've picked. It's for any sets of pairs of images that are complex compared to simple. The complex images and the simple images are going to have really different responses in primary visual cortex, but similar responses in ventral temporal cortex. And so just to remind you of where these parts of the brains are, primary visual cortex is at the back of the brain, and ventral temporal cortex, also sometimes called the ventral visual stream, is at the bottom of the temporal cortex, so on the bottom surface of your brain. Okay.
So when you record at the neuronal level, do you still get grating in the ventral temporal cortex, or is that why there's a difference? Like, are there different responses in the primary visual because it's the whole like grated on-off neuronal firing, and then is ventral temporal slightly different?
Totally. Yeah,
Okay.
exactly. So basically the question was like the neurons in early visual cortex obviously represent very different things than the neurons in ventral temporal cortex because they have things like on and off bipolar cells, and they represent curvilinear and different spatial gradients. And the ventral temporal cortex doesn't. It does this object-centered perception. So that's exactly why you're going to get these really different responses in primary visual cortex, but similar responses in ventral temporal cortex. Does that make sense? Cool. All right. And so in ventral temporal cortex, there are regions that respond that support perception of specific kinds of object categories. So there are regions that support perception of faces. So for example, if you stimulate neurons in the face region, people will report seeing a face even when there's no face, or even when they're just looking at a black box. They'll say, oh, it looks like there's a face there when it's stimulated, but when it's like a sham stimulating somewhere else, they won't report seeing a face. So that's how we know, experiments like that is how we know that this face region supports face perception. And there's also a region that supports body perception and a region that supports scene perception. And so just to orient you to these regions, we're going to call that first face region the fusiform face area, or FFA, and it's on the ventral surface of the temporal cortex. We're going to call the scene region the parahippocampal place area, and I've highlighted here where it is in the adult brain. And that body area we're going to talk about today is going to be called the extrastriate body area, or EBA. And I just told you that each of these regions support perception of their preferred category, but an interesting feature of these regions is that they also have selective responses. So all that means is that something like the fusiform face area responds much more to faces than to images without faces, whereas the parahippocampal place area responds much more to images of scenes than images without scenes. Cool. So we can use this this idea of selectivity to infer perception. So now we can ask, do infants have category selective responses in these regions in ventral temporal cortex? So while babies were in the scanner, they were watching these videos of faces, bodies, objects, and scenes. And in adult studies, what we would usually do is have like a fixation between each kind of condition, but you might imagine babies aren't going to sit very still for a fixation. So instead of trying to even get them to do that, we showed them this curvilinear baseline instead of a fixation. So all of the responses are going to be measured relative to this curvilinear, these abstract colorful scenes. And again, they're watching these movies in the mirror over their eyes while they're in the scanner. So the first question we're going to ask is there evidence for face selectivity in infant FFA? And again, the FFA is associated with face perception in adults, right? So here's the FFA on an infant brain. I've just taken the adult FFA and plopped it onto an infant brain so you can kind of get a sense of where we're going to look for responses. And in a couple of representative infants, we see face responses that are greater than object responses in the region where we'd expect to see them. But what we want to know is if these responses are going to be selective in the same way that they are in adults. So I'm going to plot the responses here on the magnitude of response relative to that baseline on the y-axis. And here's the response to faces in purple. All right, so how many of you think that there's going to be a difference between the response to faces and the response to objects? In other words, like, are we going to see the same thing we saw before, or are we going to see something that's more like the adult response? Before? Same thing as before? The adult-like response? Yeah, I wouldn't be here if we saw the same thing as before. So the face response is greater than the body response and the object response and the scene response. So this is really exciting. This suggests that these babies are representing faces as distinct from bodies, objects, and scenes in the fusiform face area. These babies are 2 to 9 months old. Sorry, I didn't say that already. So that's super excited. And our evidence suggests that there is face selectivity in infant FFA. So now we're going to move on to the scene region. So the PPA supports scene perception in adults, and we're going to ask the same question. So here's a PPA, just again from adults plotted on an infant brain, so we know where to look. There are some scene responses that are greater than object responses, which I've outlined right there. And we're going to again plot the magnitude of response. Here's the response to scenes in green. It's greater than the response to faces and bodies and objects. So evidence suggests that there's scene selectivity in PPA. But does anybody notice anything funky about this plot?
The beta's negative.
All of the betas are negative. Yep. Anything? So yeah, all of the betas are negative. But what's interesting is this scene response is not different from the baseline response. So these abstract colorful videos and scenes, there's no difference between those two categories in these data, at least. So there's an interesting puzzle like about what PPA is representing, if it's responding similarly to both these abstract colorful scenes and to like typical environment scenes. All right, so if you have ideas, tell me later. All right, so the last region we're going to look at is the extrastriate body area, or EBA. Again, this is a region that supports body perception in adults. And I'm just going to throw an adult EBA region onto an infant so we know where we're looking. And you can see that there are some body responses that are greater than object responses in the approximate area we would expect them. And here's the response to bodies. You think that infants can also represent bodies as different than other categories? Maybe they'll think bodies and faces are the same. What do you guys think? You think they'll be different? Okay. They are different, yes. Just like adults, infants' brains represent bodies as distinct from faces, objects, and scenes. So our evidence suggests that infant EBA has body selective responses. So can we measure meaningful responses to visual stimuli in infants' brains with these new coil and much more data from infants? I would say yes, we can. And we have evidence for category selectivity to faces, bodies, and scenes, which are also category selective in adults. Any questions about this section?
So in the beginning, you said that there was no difference between.
In the previous study, yeah.
Yes, so it means that they have the lower SMR. So
Yeah. Yeah, I think it's a combination of SNR and I think and data quantity. They only had a data from a small number of subjects.
Just any differences in maybe the intensity of responses with older.
Sorry, I can't hear you.
Do you see the differences between the younger babies and the older ones and how they responded or like the intensity of the response?
Hold that question. If I don't answer it after the next section, then push me on it. Okay. Yes. Wait, what's that?
Like if it is not a photo of her face, but just a painting of her face, is there any, did you try?
No, we didn't try still images for a couple of reasons. One, babies don't like still faces, and we wanted to be able to collect lots of data. So having them look at still faces would have been hard. And the other reason, well, I mean, those are both reasons. Babies don't like still faces. Collecting images of still faces would have been hard. I think that that's actually an interesting question. If still faces and moving faces would evoke the same response in FFA, my guess would be no. In infants, in adults they do, but my guess would be no. Yeah. Any other questions?
So you chose the media with the patterns as the baseline. So what about the pure dark image as the baseline? But there would be any difference like the same understanding the region.
So your question is, what if we used a different baseline?
Yes.
Okay, so for the same one. In Ben's paper, they used scrambled scenes as their baseline, and there was still no difference between the response to scenes and scrambled scenes. Yeah. Any other questions?
If you used a cartoon of a non-human face that was moving compared to a human face, has anyone done that? Compare like the effect of it being like a human being?
Nobody's done that in infant FFA. People have done that on infant STS and infant MPFC, but I want to hold it for later. That's okay. Great. FNIRS is good for STS and MPFC, whereas FFA you have to use fMRI, so it's just harder to get a lot of different. Somebody did do sheep faces though, and they got it. Okay, so next, do responses in temporal and prefrontal cortices emerge sequentially? All right, so here's a picture of a little baby staring at their father, and it would look like this baby is doing more than just perceiving a face, but inferring a social relationship, perhaps. And in fact, we all do more than just perceive faces, right? These are not any people you know, but you might be able to make inferences about these people, about whether or not you might like them or whether or not you know them, and what kind of person you think they are just by a glance of their face. And so perhaps it's not surprising that there are many regions in adults' brains that also respond to faces, but for different reasons. So we've already talked about the fusiform face area, which I told you supports face perception in adults, but there's also the superior temporal sulcus, which responds to faces but supports social perception. So observing two other people engaging and the kind of engagement they're having with each other, that's going to evoke responses in STS. And then the medial prefrontal cortex, or MPFC, has a face responsive region that computes social inferences. So like, do I like that person? What do I think about that person? What does that person think about me? Those kinds of things are going to evoke responses in MPFC. But just like with perception, there's an interesting feature of these regions where if you only measure responses to visual categories, you also get face-selective responses. So here, these are adult brains, and you can see the FFA, SDS, and MPFC all look like they have face-selective responses, even though we know they're doing very different things in adults. Okay, another interesting feature of these regions is that they are feed-forward. So what that means is that temporally, the fusiform face area responds to faces earlier than the STS, which responds to faces earlier than the MPFC. And so I don't focus too much on these specific numbers. These are the numbers from this one study, but there have been other studies that find a similar progression, but different numbers. Sometimes the FFA is like 150 milliseconds versus 400. But the point is just that the response in FFA happens first, the response in STS happens second, and the response in MPFC happens third. And because you all are really interested in infants' brains, you know that different parts of the brain develop at different paces, right? So regions that are closer to sensory cortices are going to expand and myelinate earlier than regions that are farther away from sensory cortices, and especially higher order association cortex. And so here I'm just showing two different measures. The one on the left is Fleisching. I don't know if you guys are all familiar with that, but that's just like the rate of myelination. And you can see that myelination happens faster in FFA than it does in STS and MPFC. And this plot on the right is just about cortical expansion. Again, happens faster in FFA than it does in STS and MPFC. So these kinds of results from adults and from anatomical developments have led to something called the serial hypothesis of cortical development, which supposes that there's something like a face detection mechanism that's subcortical and fast and genetically predetermined, and that orients infants to faces, and all of that orienting to faces leads to the development of face-selective responses in FFA, so babies learn to distinguish between different faces, and once they have a fully face-selective response in FFA, then they're going to develop face responses in STS, where they're going to learn about inferring social interaction, so like who's their mom talking to and things like that. And then last, there's going to be a face selective response in MPFC, because this is the last region to respond to faces, and because it myelinate, anatomical development happens so much later. Does that make sense? It's pretty intuitive. It follows a bunch of things we know about the brain already, but there's an alternative hypothesis that we call the parallel hypothesis of cortical development. And this suggests that each of these regions are going to develop face-selective responses at the same time, maybe due to bidirectional connections with the thalamus, or maybe due to differential inputs from different subcortical structures, which is also supported by anatomical studies. Does that make sense? So we have two hypotheses, the serial hypotheses and the parallel hypotheses of cortical development. So now we're going to capitalize on these. We're going to test these two different hypotheses using specifically face responses in FFA, STS, and MPFC. So the specific predictions are going to be for the serial hypothesis, the FFA face-selective responses will emerge first. STS face-selective responses will emerge only after FFA has face-selective responses, and the MPFC face-selective responses will emerge last. So that's the serial hypothesis, whereas the parallel hypothesis predicts that as early as infants have face-selective responses in FFA, they're also going to have face-selective responses in STS and MPFC. All right, so now let's ask if face responses emerge sequentially. I'm just going to take a drink of water.
Could it be possible that in different infants, depending on their early environment, they would go down one or the other?
Like two different routes of cortical development?
Like it would be like a balance between them both. You could shift in one direction depending on like how much exposure you have to your parents, how much they interact with positively, etc., etc.
So what we're talking, it's in typical development, I think, I mean, I guess it could be, but it could be. I don't know. Yeah. Any other questions? All right. So here are our three brain regions we're talking about here. There's also other brain regions that you might want to think about. So maybe if you start talking about a bunch of different brain regions, that could be the case too. But let's just talk about these three for now. So I've already showed you data showing you that these 2 to 9-month-old infants have face-selective responses in FFA. But the serial hypothesis predicts that the face-selective responses in STS and MPFC won't develop until after FFA. So it's possible that there won't yet be face-selective responses in STS or MPFC. Here's the response to faces in STS. Does anybody think that we're going to find selectivity in STS? One person, maybe? All right. Cool. So you guys are like really bought into the serial hypothesis. That's fine. It's great. The face response is greater than the body response, and also the object response, and also the scene response. So there are face selective responses in STS, but it's still possible that we could find evidence for the serial hypothesis in the MPFC. So we're going to look there next, right? Because the MPFC should be the last region to develop face selective responses.
Did you use specific agents here?
These are 2 to 9-month-olds.
Okay.
Yeah.
I think if you were to like stratify by month of age.
I guess we'll have to wait and see. Yeah. All right. So does this mean that you think there'll be face selective responses in MPFC? Now I've brought you over to the parallel hypothesis. Great. Anybody else want to be on the parallel hypothesis side? Couple people. All right. Yes. So here's a face response. It's significantly greater than the response to bodies, objects, and scenes. So far we don't have evidence for face selectivity, but as you astutely realized, these babies are 2 to 9 months old. That's a really big age range. There's a lot of change that happens between 2.5 to 9 months. And so if we really want to know if face responses emerge serially, maybe we need to just look at the youngest infants, those that are only 2.5 to 5 months old. Another benefit of looking at just these younger infants is that even with FNIRS, nobody has looked at face responses in babies this young. So we are contributing to the literature even more by looking at babies' responses to faces in this really young age range.
Did you limit yourselves to a certain number of minutes for each baby? Or
No.
did you just kind of throw everything in?
Everything. Yeah. Yeah. We want it to have as much power as we could. And we just did, and this is actually only a median split by age. So we just took the youngest half of the babies. Yeah.
I was wondering if Rebecca's got the hyper intelligent baby with 2 hours of data, whatever age.
Well, those data aren't here.
Oh, okay.
Yeah, these are just the data I collected. Those data are like part of Ben's paper and not here represented here at all. None of these babies had a parent in the lab.
Oh, okay.
Yeah, so I mean, there still could be some hyper intelligent babies mixed in, just not Rebecca's.
Okay.
Cool. All right, so we're going to start with the FFA and these 2.5 to 5 month olds. Here's the response to faces. As you can see, it's a smaller magnitude than anything we've seen before. So maybe if we're going to find evidence for the serial hypothesis, this is exactly what the age range we need to be looking in. But the face response still is significantly greater than the response to bodies, objects, and scenes. So again, this seems like a really good place if we're going to find evidence for the serial hypothesis of where it would be. So let's look at the STS. Y'all ready? Yeah? All right. Here's the response to faces. I like building up the like. It's significantly greater than the response to each of the other categories. So we don't have evidence for the serial hypothesis here, but what you might be saying to yourself is, I remember I looked at those plots earlier. The FFA and the STS are both in the temporal cortex. Maybe they do develop at the same times, but we're definitely not going to see anything in MPFC, right? So if you are like super bought into the serial hypothesis, that's something you might say. So let's check MPFC. Here's the face response. It is greater than the response to each of the other categories. So we don't have, wow, face selectivity in all three regions. It's very exciting. And so we don't have evidence for the serial hypothesis, but we do have evidence for the parallel hypothesis. And for those of you that are naysayers, there is behavioral evidence for social inference in infants. Tongue twister. So there was a study with macaques where they were deprived of seeing other faces for the entire infancy. And then they got to choose how much time they would spend with different types of faces. And one type of face they got to see was an infant face, and one type of face they got to see was a threatening adult face. They spent much more time choosing to look at the infant face than they did the adult threatening face. They'd rather not look at anything than have to look at the adult threatening faces. This is a cool study, but it turns out it's maybe not true, but I still think it's a cool study. Have you all heard of the helper-hinder paradigm? Infants like shapes that help other shapes more than shapes that don't help other shapes. So getting at your little, what if it's just cartoon character faces? There seems to be some evidence for that. Infants like imitators over non-imitators. Infants like faces that speak their native language more than faces that speak a foreign language. Infants like faces of a stranger that is singing the same song their caregiver sang more than a face that's singing a very similar but different song than their caregiver sang. And then infants also expect people that they saw their mom interact with to call their name. So there's lots of reasons to suggest that maybe MPFC is actually representing social inferences in really young infants. Okay, so do responses in temporal prefrontal cortices emerge sequentially? I hope I convinced you that they emerge in parallel, suggesting that for infants, faces are more than just a special perceptual object. So anybody have questions about this section?
So this may be indirect the question. So what I'm thinking about is about the autism spectrum. They seems to be like the social cues. So
Wait, did you say you're thinking about the auditory spectrum?
the autism spectrum.
Autism spectrum. Okay, got it.
So they seems to make of the social cues of them. So maybe interesting to see.
Totally. I'm trying to write up a grant right now for the Simons Foundation because I think it has lots of implications for autism. Yeah. Any other questions?
If mum or dad is in the scanner, do you think that would inflate the signal for looking at faces because there's a face nearby in addition to the stimuli?
I mean, if the baby was looking at their mom's face during non-face things, maybe, but the parents usually have like their face down. It's not very comfortable.
I guess that adds to the baseline anyway, so you definitely would still, with it, all of it being comparative, there's still like a higher face thing. I was wondering if it kind of enhances the paradigm.
I don't think so, because it would have to happen during only the face blocks.
Oh, okay.
Right? And the parent isn't going to know which are the face blocks, right? And so if a baby happened to look, but also if the baby happened to look at their mother, then I had a coder that was outside the scanner that would tell me that the baby's not looking at the stimuli, and we wouldn't include those volumes in the analysis. Yeah. So ideally, that's not a problem. Yeah. Any other questions? Great. Okay, now I'm going to move to audition. That's why I thought you were, I was really excited when I thought you said auditory, but this section is pretty short. Yeah, so just can we measure meaningful auditory sounds in infants' brains? So these are just two cochleogram plots. So a cochleogram is just a measure of how much power there is at each frequency and in each time point. It's an approximate representation of the activations to the cochlea. And those activations are sent through the auditory system to primary auditory cortex. Again, there's going to be these person-centered versus object-centered sounds. So I'm going to just play these two sounds for you. Here's the first one. Did everybody hear that? It was only the first 5 seconds. Okay. Did it sound familiar to anybody? Like something you've heard before? No, it was kind of annoying, like scratchy, maybe. I don't know. It grates on my nerves. So that second sound sounds really familiar, right? So it's a mother talking to their baby about a monkey, but they have pretty matched spectral cochleograms. So an interesting feature about doing something like this, as you might be expecting already. Oh, sorry. So we'll call this one infant-directed speech and that one model matched speech. And again, we'll talk about this person-centered versus, so the person-centered sound would be that's infant-directed speech, regardless of what direction you're hearing it from. And that's modeled matched speech, regardless of what direction you're hearing it from, but they're both activating primary auditory cortex similarly. Okay, so in an adult speech region, the response to speech is going to be greater than this response to the model matched speech. And now I'm going to play 2 more sounds. That was just the first 5 seconds. Does that sound familiar?
Bad flute player.
A bad flute player, yeah. Anybody else? Okay, this is the original sound. Very good flute player. All right, so I'm going to call that first the flute player, the monophonic instrumental lullaby. We picked instruments that play in the same frequency range as infant-directed speech and that have sounds only coming from a single source. So it would be more like the human voice. And then we also did the model matched music stimuli. And in the adult speech region, the response to speech is still greater than the responses to music and model matched music. But we're also going to look in the adult music region, and we're going to see that the responses to music is significantly greater than the responses to these other categories. So you might be able to guess at this point that we're going to want to do this in infants. So let's do it in infants. These infants are 2 to 9 week old infants, so much younger, but they're also sleeping, so they're not awake. Here is the response to speech. How many of you think that we're going to find speech selectivity? Anybody say no? Okay. We actually don't find strong evidence for speech selectivity. The response to speech is greater than the response to music, but the differences between speech and model matched speech and model matched music are not strongly significant. They're actually pretty weak. There is a difference there. Yeah.
So I noticed that the infants here is much younger than the 9 weeks old.
Sorry, what?
So for infants here is 9 weeks old extent of the 9%.
Yeah, these are 2 to 9 week old infants, yeah. And they're sleeping. So I think that's an important part of this. So it's kind of confusing, but it's also kind of interesting. I want to replicate this in awake infants to see if like the fact that they're sleeping is part of what's going on here. But now let's look at the music region. All right, here's the response to music. How many of you think it's going to be selective? How many say no way? If speech isn't, why would music be? It is selective, which is also really surprising. So this is not exactly what we thought we would find, but it's a cool finding. Again, I want to follow it up in awake infants. It's the first time anybody's measured music selectivity in infants, so it really needs to be replicated, but it's cool. And it shows that we can measure meaningful responses in infants' brains to music, to auditory stimuli. Cool. And so future directions. I guess I can go through future directions, or we can go on to questions if you guys want, because I think it ends in like 8 minutes. Is there a preference? What time does it end?
11, but a little bit of wiggle room.
Okay.
I can go for future directions.
Okay, great. Oops. Yeah, that's a great question. Um, I did try to scan adults sleeping, and some of them did fall asleep, or at least got really drowsy. And but I didn't measure their sleep state like the proper way. And those data I showed you are from those adults. Um, there's a lot of mixed findings in the literature about whether or not adults perceive these categories as selective while they're sleeping. So I don't know that it's a solved problem, but it is, I'd like to solve it at least for these stimuli, because it'd be interesting to figure out if infants are learning like speech sounds and supporting language development while they're sleeping or not, right? So, but it's kind of mixed. Yeah. Any other questions?
Fair in adult and infant sleep because stages are different. Like that young, just like 2 weeks old, sleep stays, they have not like proper sleep stays. So
Totally. Yeah, I think so too. Yeah, that's also why I hope to like start like just look at the awake data first, and then if it's similar, then maybe dig in more. But if you get like reasonable responses or like something that seems reasonable in awake infants, then we can say whatever's going on in sleep and infancy versus awake states is different. And then maybe dig into it later. I probably need to like collaborate with a sleep researcher because that's not my my domain. But yeah, I think that's an important point. Okay, so one future direction is to like figure out this speech thing, which we kind of already been talking about. But what's interesting is that the auditory network and the language network both develop in utero. So all of these connections are already established by the time an infant is born. The auditory network anatomically does develop earlier than the language network anatomically, but the important feature is that they're both in place prior to birth. And also in neonates, you can identify the language network using resting state data, so, which is a indirect proxy of anatomical connectivity, right? And so one thing that would be fun to do is again test these ideas of the serial hypothesis in the language domain. So specifically, people have said for the serial hypothesis that speech selectivity will emerge first in non-primary auditory cortex, then temporal language regions will develop speech selectivity after non-primary auditory cortex, and then speech selective responses will develop in prefrontal cortices last. But alternatively, maybe like with faces, these regions are going to be developing speech responses at the same time in development and then taking on different representations as they learn more. Okay, so that's a fun future direction. Another future direction I've been thinking a lot about is these visual responses. They're very cool, but they also leave a lot of open questions, such as like, are infants really representing faces? Because I didn't do any low-level visual controls. So they are these, which is some of the questions that you guys were all asking about too. There's a new model matching algorithm that can take videos and kind of warp them so that they have the same low-level visual statistics, kind of like we did with the auditory stimuli. So we can measure responses in infants' ventral temporal cortex to see if they have person-centered vision, which would predict a similar response in an FFA. Would it predict similar responses to the faces and the model-matched faces? Whereas object-centered perception would predict a strong response to faces and not the other categories. Does that make sense? Another open question, though, is I showed you that STS and MPFC have face-selective responses, and I inferred different kinds of functions for them based on adult brains, but I actually don't have any evidence that they're doing any different functions. So I want to try to measure those responses too. So one hypothesis could be that all of the regions are just responding to faces and no social content. Another possibility is that a face region is representing the self-relevant information. So in this kind of condition, you would expect a strong response to the caregiver, but not to like strangers interacting. And then for social interaction, you might expect a strong response to strangers interacting, but not to people that weren't interacting. So that's a fun future direction I'm hoping to get to. But the thing I've been really interested in, and I've spent a lot of my postdoc thinking about and trying to learn how to do in adults, is trying to understand how whole brain networks support cognitive development. And so there's a prominent theory in cognitive development called core knowledge, which suggests that knowledge domains are in discrete domain-specific clusters. And it's somewhat supported by neuroscience data that shows that the brain actually does have domain-specific networks that are distributed across the cortex. So, for example, this is the theory of mind network. It has different regions in different parts of the brain, and it responds a lot when people are thinking about other people's thoughts and feelings, but it doesn't respond when people are thinking about other people's actions or thinking about other people's bodily states, such as thirst and hunger or pain and pleasure. It really seems to be specialized for thinking about other people's thoughts, right? So one might ask, does the theory of mind network respond to people in infancy too? And importantly, does the theory of mind network distinguish between faces and bodies in infancy? We know in adults that it does, but will it in infants? Because if you think about a face, oftentimes a face is kind of the gateway to the mind. It helps you think about what other people are thinking or what they might be feeling, right? And so even though infants don't have rich theory of mind capacities in the way we do, they might have some rudimentary ones. And so I already showed you data that the MPSE here and the STS here are responding to faces selectively to faces in infants. But I'm also going to show you that two other regions in the theory of mind network, the medial parietal cortex and the temporal parietal junction, are also responding more to faces than other visual categories. So this suggests that the entire theory of mind network is not only in place in infancy, but also responding to faces in infancy, which leads to like really interesting questions about how these representations really in infants lead to the higher level cognitive theory of mind representations that we have as adults, or even like a 5-year-old has. So I'm super interested in trying to push this line of research forward. And then, but it leads to the question of like, how does the brain support within domain learning? So we have this theory of mind network, but like, how are those representations updated? Sure, there's lots of information from the environments coming in, but what does that mean? So I'm going to go back to the language network just because it's an easier way to kind of like show you my thoughts. So this is just like the frontal and lateral language regions. Each of these regions project to the same part of striatum. So different regions that are part of the same network project to similar regions of the striatum. We know that from lots of anatomical studies with macaques. So one hypothesis that I have is that the striatum integrates information across nodes within a network. So theory of mind and language representations aren't going to the same place. Only theory of mind and theory of mind representations are going to the same place. And then that information is getting integrated and sent to the striatum, and then integrated in the striatum. And then the striatum kind of sends like a go, no go signal back to the cortex through the thalamus saying, should you update this representation or should you not update this representation? And I think this whole process is going to support language acquisition within the language network, and that it is happening simultaneously in different networks that have different specializations. So that's a hypothesis I'm trying to develop. If you guys have ideas about it or want to talk more, please let me know. And that's it. So future directions, I showed you some ideas about the language network, some ideas about testing social interaction perception, some ideas about how language networks develop. And with that, I'll say thank you for your attention, and to my advisors and their labs and all of my collaborators and the babies in my research.